Dear students, welcome to my video tutorial class. Myself Minakshree from the Department of Biotechnology, Darrang College. Today I will discuss the topic cell adhesion molecule. The contents of my video tutorial class on cell adhesion molecules are plasma membrane, cell adhesion molecules, structure of cell adhesion molecules, molecular interaction involves CAMs, classification of CAMs, and different classes of cell adhesion molecules. Before we go into cell adhesion molecules, let's just refresh our knowledge on cells and plasma membrane. We all know cell is the basic unit of life, and as an independent unit, cells can perform all the functions necessary for living organisms, whether it is in unicellular organism or in multicellular organism. In case of multicellular organisms, cells forms tissues to perform various functions. Similar cells, they can form tissues with the help of the molecules which are present on the cell surface, and then they perform specific functions. They adhere to each other and communicate within a tissue so that the functions can be carried out in a very smooth manner. You can see in the diagram, this is a cell, and within the cell there are different organelles. We can see the nucleus is there, then this is your endoplasmic reticulum, mitochondria, Golgi body, and various other organelles are there. This is a cell from multicellular organism. And this cell has a limiting membrane which is separating it from the surrounding environment. And this membrane is called plasma membrane. Plasma membrane is composed of phospholipid molecule and proteins primarily. Phospholipid molecules are arranged in two layers and protein molecules are embedded in it. Some of the protein molecules are just bonded with the phospholipid molecules on the outer side. Some of the protein molecules can actually span the phospholipid layers, through which some of the molecules which otherwise cannot pass through the plasma membrane pass through these channels and reaches the interior of the cell. So this plasma membrane separates the cell interior from the exterior. It has two areas. One is your extracellular area and one is your cytoplasmic area. Extracellular areas, it has got glycocalyx and some of the proteins, and these proteins are called surface proteins. And in the interior side, it has got cytoskeleton structures. Some of the integral protein reaches to the cytoskeleton structures and it connects with the exterior of the cell through channel proteins. Cell adhesion molecules are those cell surface receptor proteins which are present on the surface of the plasma membrane, and it promotes cell-to-cell interactions and cell-to extracellular matrix. And these are important for various biological processes like tissue-specific functions, embryonic cell migration, immune system functions, wound healing, and intracellular signaling pathways. Let's see the structure of the cell adhesion molecules. All the cell adhesion molecules have three major domains. One is extracellular domain. We can see in this diagram, this part is extracellular domain. And one is transmembrane domain. So this is the plasma membrane, and the domain which can span through the plasma membrane is called transmembrane domain. And there is one domain which is on the interior part of the plasma membrane, that is in the cytoplasm area, that is called cytoplasmic domain. So these three domains are important for various functions of the cell adhesion molecule. The first domain, the extracellular domain, is primarily involved in establishing the interactions with CAMs from the other cells. Transmembrane domain connects the extracellular domain to the cytoplasmic domain, and cytoplasmic domain helps CAM molecules to interact with the other proteins which are present on the cytoplasmic area. There are three different types of molecular interactions in which CAMs are involved. Number one is homophilic binding. Homophilic binding facilitates interactions between similar CAMs. So the CAM present in one cell surface can interact with CAMs present in another cell if they are similar, that is your homophilic binding. Like cadherin can interact with another cadherin present on the cell surface of the another cell, and that will be homophilic binding. If a cell adhesion molecule of one type present on one cell interact with another type of cell adhesion molecules present on the second cell, that will be heterophilic binding. So here the cell adhesion molecules are not similar, but they can interact. And the third type of molecular interaction is present, in which the cell adhesion molecules are not able to interact on their own, but they need some help of the linker molecules, and with the help of these linker molecules they can establish contact and communicate and perform interactions. And this is called binding to an extracellular linker molecule. Mostly linker molecules are laminin in this case. Let's see the classification of cell adhesion molecules. There are 4 different classes of cell adhesion molecules: cadherin, selectin, immunoglobulin superfamily, and the integrins. Cadherin is one of the largest family of cell adhesion molecules. They are dependent on calcium for their functions. It plays an important role in cellular communications by forming desmosomes. It mediates intercellular adhesion in epithelial and endothelial cells by calcium-dependent homophilic adhesion. It plays a critical role in cell adhesion, embryonic development, and tumor metastasis. And failure of cadherin-mediated cell-to-cell adhesion cascade may lead to breast cancer. We will see the structure of cadherin molecule. So here in the picture you can see this is cell 1 and this is cell 2. And from the cell 1 you can see this is one cadherin molecule, and from cell 2 this is another cadherin molecule. It has a short cytoplasmic domain, then a short transmembrane domain, and a long extracellular domain. Extracellular domain contains repeated unit of cadherin. These are EC1, EC2, EC3 and EC4, and these repeats have got calcium binding site, and it interact with the cadherin molecule interact with specific cytoplasmic protein. They are called catenins, and which are very important for their activation. Next class of cell adhesion molecules is selectin. These are carbohydrate binding cell adhesion receptors, and there are 3 different forms: L-selectin, P-selectin and E-selectin, and it plays a very important role in defense mechanism, leukocyte migration, inflammation processes, etc. Let's see the structure. It has again long extracellular domain, and that long extracellular domains, in the outermost part there is a C-type lectin domain. And this C-type lectin domain determines its specificity. Then it has an epidermal growth-like factor. Then there are some consensus domain. Then a short transmembrane domain which is connected to the cytoplasmic domain. This is the structure of P-selectin. The next cell adhesion molecule class is immunoglobulin superfamily. This is the largest and most diverse family, and it exhibits both homophilic and heterophilic binding. It can adhere to the same molecule, or it can adhere to a different cell adhesion molecule. It involves in recognition, binding and adhesion processes of the cell. These are different classes of immunoglobulin superfamily cell adhesion molecule. There are 4 different classes: intracellular adhesion molecule, intercellular adhesion molecule, vascular cell adhesion molecule, and platelet endothelial cell adhesion molecules. Basically, it is involved in development of nervous system, cytoplasmic assembly, synaptic composition and plasticity. The last one is integrins, one major class of receptor within the extracellular matrix. It mediates cell to extracellular matrix interactions with the help of different proteins like collagen, fibrinogen, fibronectin and vitronectin. These are formed by one alpha and one beta subunit, and they are various combinations of this alpha and beta subunit which can give rise to 20 different types of integrins at the cell surface. And main functions of integrins are cell migration, ECM binding, cell signaling, cell cycle regulation, etc. You can see in the diagram the structure of integrin. There is an alpha chain, there is a beta chain on the extracellular domain. Then there is a long transmembrane domain, and then there is cytoplasmic part where again there is alpha and beta site. And then they have got binding site for one protein called talin, that is a cytoplasmic protein. Let's see how it functions. Integrin functions in two different modes, outside in signaling and inside out signaling. In outside in signaling, the stimuli is from outside, and in inside out signaling, stimuli is from inside. So in outside in signaling, the ligand binds to the integrin in the extracellular domain, and then it changes the conformation of the integrin molecule, which leads to clustering of integrin, and then you can see the result on the interior side of the cell. The functions involved in outside in signaling are cell polarity, survival and proliferation of the cell, cytoskeletal structure changes, and gene expression. In case of inside out signaling, where the stimuli is from inside, so there is a protein called talin. It has talin binding site on the cytoplasmic face. Talin binds to the integrin, it changes the conformation, and then it leads to a cascade where the result you can see on the outer side of the cell. And this type of signaling is important for cell adhesion, migration, and extracellular matrix assembly. So these are two different types of signaling ways. Let's just recap what we have learned so far. There are 4 different types of cell adhesion molecules which are important for cell to cell interactions, and that interaction is important for performing a function as a tissue. Unless they communicate and interact with each other within the tissue, they cannot perform the biological functions. And to perform this biological function, they are dependent on these 4 classes of cell adhesion molecules. Number 1 is cadherin, number 2 is IC family, IG superfamily, then integrin and selectin. And all of them have got 3 different domains: extracellular domain, the transmembrane domain, and the cytoplasmic domain. And all 4 classes have got different extracellular domain. Cadherin has got calcium binding site in its extracellular domain. IG superfamily has got a long extracellular domain with disulfide bond, and this looks like immunoglobulin. Integrin have got one alpha and beta chain, and they connect and perform different functions with the help of conformational changes. Then for selectin, it has a lectin domain and epidermal growth factor domain, and there are some consensus repeat on the extracellular domain. This is all on cell adhesion molecules. Thank you for watching my video tutorial class. These are few selected references. Thank you.